So a question for Scott. Scott, do you know if that electrofishing is as accurate? It probably would be as accurate as the eDNA, just as a question off the top for
That's a pretty complicated question.
Oh, okay.
And I would say I would say it's more accurate because electrofishing has been tested and used all over the place for many, many years, and there's a lot of studies behind it. eDNA is just barely starting to be explored to measure abundance. And so maybe it'll be better someday. We just don't know. But I'm actively involved in some of that research, and I'm gonna go through the two science examples I'm gonna give are related to that. So
Excellent.
stay tuned.
Okay. That's great. I haven't done electrofishing myself, but a number of people I've met through my stint as president of the Jemseg Grand Lake Watershed Association and do it regularly. So it seems quite a common thing. My name's Brad Nickerson. I'm a member and past president of the Jemseg Grand Lake Watershed Association. We are very grateful to Scott Pavey for taking the time to prepare this webinar and for sharing his deep knowledge of and passion for environmental DNA with us this evening. Our watershed covers 3,950 square kilometers. That's about 70% of the size of Prince Edward Island. It is a sub-watershed of the much larger 55,000 square kilometer Woolastook or St. John River watershed. And Grand Lake at 171 square kilometers is the largest freshwater lake in the Maritimes. If you're not already a member of our association, we invite you to join. Membership costs $10 per year and supports our activities, including the organization and hosting of this webinar. You can just visit our website and click on the blue Join Us button in the upper right corner. We welcome your questions. Please use the chat window, as you've been doing, to introduce yourselves, to ask any questions as you think of them, as they come up. And then I'm going to keep track of every question in the order it appeared. And I can actually go back into the chat list and see the questions in order. And after Scott's presentation, he's going to answer those questions. Hopefully we'll have enough time to answer everybody's questions. If not, we'll email you the best answer we can come up with after the webinar ends. I'm going to just introduce Scott's background. He has a very interesting background. Dr. Scott Pavey is a full professor at the University of New Brunswick in St. John in the Department of Biological Sciences and Canada Research Chair in Aquatic, Molecular Ecology and Ecological Genomics. He leads CRI Genomics, a state-of-the-art lab, uses modern genomics techniques to address ecological questions. Projects fall into three broad categories. I should mention his lab is in St. John, so anybody that's interested can interact with his lab directly. First category is population genomics, which is genotyping individuals and populations to understand migration and adaptation. Second, transcriptomics, which is measuring gene expression to determine physiological states and responses of animals. And the third, which we're going to hear about tonight, environmental DNA, which is using residual DNA suspended in water to determine presence and abundance of species. The ultimate goal of Dr. Pavey's research is to develop the tools and understanding needed to conserve Canada's vital freshwater resources. Okay, turn it over to you, Scott.
Great. Well, thank you very much for that very nice introduction. And I'm very happy to present here today. First, I just wanted to say have one slide about my department. So it's the Department of Biological Sciences at University of New Brunswick in St. John. And we're overall like a marine and coastal department. But there's a lot of freshwater work as well. So it's a great department. I love it here. And we have really serious researchers that cover ecology, oceanography, genomics, aquaculture, physiology, environmental chemistry, and bioacoustics. So that's just an introduction to the department. This is where my facilities are, the CRI building. The CRI is a institute that actually has researchers in both of the campuses. There's a lot more researchers at the UNBF campus, but the only CRI building is in St. John. And so this was a $5 million facility that was built in September of 2006. The building infrastructure, so there's 2 genomics labs, and one is actually for a professor that's about to arrive this July. There's an ecotoxicology lab. We have, in the basement, we have live fish husbandry and experimentation, and we have plumbed in salt, brackish, and dechlorinated freshwater. So it's a really nice state-of-the-art facility. And then offices of a bunch of the department's professors. So then CRI Genomics is my lab, and it is a state-of-the-art genomics lab specializing in aquatic molecular ecology and ecological genomics. So the mission is to study DNA in nature to help solve problems. And we have big research programs that focus on striped bass and Atlantic cod, as well as aquatic invertebrate microbiotas. We have about $1 million of infrastructure in the lab, and I've acquired about $2.5 million in external research funding so far, and I've trained approximately 32 highly qualified personnel. I started in 2015, so it was about 7 years ago. So that's an overview of myself and my lab and our department and our facilities. So now we're going to get to the big show, because today I'm here to talk about environmental DNA. And DNA is falling off of animals all the time, including humans. In every single cell in your body is a complete copy of your DNA. So it's each cell is only using a very small subset of that complete copy, and a brain cell would be using a much different part from a muscle cell, etc. But every cell has a complete copy of the organism's DNA. So when cells are shed, especially into water, they are held in suspension, sometimes for quite a while. And so we can exploit that with environmental DNA analysis to identify species that are in that lake, ocean, or river. And this has been used mostly for vertebrates. I would say by far the largest amount of the research has been on fishes, but certainly reptiles and amphibians have also been used. Their environmental DNA has been used to detect them. So I have a couple active graduate students that are working on environmental DNA studies. I'm actually a participant in a Genome Canada grant that's called GenFish, that's got professors from coast to coast and all the way from here to BC, although I'm the only one participating in the grant in the Maritimes. So I have a lot of that goes to funding students to do projects. And so I'm just going to introduce quickly 3 of my students. Rena Gajoli, she's developing eDNA assays for freshwater fishes found in New Brunswick. And she's a master's student. Jagger Waters-Gray is now a PhD student, and he's also developing assays for Canadian fish found in the Maritimes. He's also doing some pretty high risk, high reward projects as well. That is really trying to see how far we can see how much information we can get from eDNA. Like we could possibly get eDNA to count individuals using microsatellites, which are just highly variable regions of the genome. That's the same that is used for human forensics. And he's also attempting to use environmental RNA as a surveying tool. RNA is just like the middleman. So you have DNA, and then to make a protein out of DNA, there's an intermediate molecule called RNA, and that is very fragile and degrades pretty quickly, but there might be some pretty neat things that we can do by detecting environmental RNA in the environment. And so he's pursuing those projects for his PhD thesis. And then Jake Riker is another master's student, and he's using environmental DNA specifically around the Mactaquac Dam to monitor the success of the fish passage and usage of the habitat above the dam. And that's all part of another big project named MAZE, or Mactaquac Aquatic Ecosystem Study, which is led by Alan Curry at University of New Brunswick in Fredericton. So those are my active students. I also have Melissa Morrison that graduated about 6 months ago, but I'm actually going to present some of her work in the science part of this presentation. All right, so the eDNA process. Jake Riker made these slides, the last person I just introduced, and so I thought they were very nice slides, and I've stolen them with permission. So the first part of the eDNA process is to collect water. This has to be done in a fairly careful fashion because we're dealing with such small quantities of DNA. Anytime we're studying environmental DNA, it's very easy to contaminate environmental DNA. So for example, you need waders that are specific to environmental DNA, in the sense that you couldn't use waders if you're actually handling fish and you were getting fish slime on the waders. You would not be able to use those for environmental DNA studies because your waders would emanate an eDNA signal. So you have to, there's a bunch of things to think about like that. We wear gloves in the field, and often we just collect the water in 1 liter bottles. We do have the capability of in-field filtration as well, but our standard is just to collect water in bottles and then to keep them cool and transport them to the lab where filtering takes place. And so as long as they're kept cool and in the dark, you've got like a decent 24-hour window before the eDNA in your 1 liter bottle starts to degrade. And so then they're taken to the lab. We use this filtering apparatus right here, and each of the 1 liter samples is put into these cups, and then a vacuum pump draws the sample through a filter, which is right in this area, and the DNA in suspension gets trapped on that filter. So then the next step is we take that filter and cut it in half. Oftentimes we'll extract one of the halves and then archive the other half just in case something goes wrong. And we then extract the DNA off of the filter. So there are various commercially available kits that we use for this. And we have again, because contamination is such a big issue with eDNA, we have our lab set up in kind of the ideal manner, in the sense that we have a separate room for every step in the process. So this filtration room is a different room from the room we extract in, and that's a different room from the qPCR prep, which I'm going to go into next. And so we have everything spatially isolated, and that's to reduce the chance of contamination. But when the extraction process is done, what you get is a small tube with some buffer, and the DNA is dissolved in this buffer. That's the end result of the extraction. So then the fourth step of the process is qPCR, or Quantitative Polymerase Chain reaction. And so this is when we actually work with the DNA. So more or less, whenever you want to study DNA, the first thing that you have to do is make many copies of the region or regions that you're interested in. And so environmental DNA is no different, but the region that we're interested in with environmental DNA is the barcode region, which has the right amount of variability so that within a species, it's generally the same, but between species, there's usually some fairly pronounced differences. So the barcode regions of a species are what we focus on. That's what you want to identify species. And we do barcoding as well in the lab. Sometimes people submit tissues to us, like somebody wanted to know, you know, there were these little larvae that looks like musky larvae that were downstream of the St. John River, but they wanted to know if they're musky or chain pickerel, because they're indistinguishable, you know, when the larvae are that size. And so they submitted those tissues to the lab. We barcoded using very similar techniques. But the qPCR, so this is the DNA right here. So DNA is double-stranded. And then our lab, or the literature, designs these molecules here, the forward primer and the reverse primer, that have an exact sequence match with the species that we want to detect. And then there's actually a third molecule that's designed called the probe that also has an exact complementary species for an exact complementary sequence for the species that we want to detect. And this causes fluorescence. So this is a fluorescent molecule that's attached to this little short segment of DNA. And then when replication occurs, that fluorescent molecule is freed up and actually starts fluorescing. So then you can tell that your species DNA that you want to test for was present in the sample if you start getting this fluorescence when PCR is occurring. And this is how that fluorescence looks. And so more or less, this chemical reaction, a PCR goes through about 40 cycles where it generates the DNA if it's present. And so this is the number of cycles right here. So it goes up to 40. And so at the very beginning, there's no signal at all. But then at about cycle 12, you start to see a signal. And so this would tell us that whatever species we tested for was in that environmental DNA sample, because we're getting lots of signal. So this process and these molecules are species specific. So you have to have a species specific assay, and then you test that eDNA sample. Was there this species in it? And then you can use that same sample and test to see if there's more species, but it's one species at a time. So that is the basic eDNA process. And there is another option, which is metabarcoding. And this one is a little bit different. So with this one, instead of species specific molecules, it's actually universal primers that amplify the barcode region, but these primers sit down in a spot that's the same amongst very wide groups of taxa, like all teleost fishes, for example. And then after that is done and you amplify all of the species that were in your eDNA sample, we use next generation sequencing to get sequence information for that sample, and we can use those sequences to actually identify all of the species in one shot that were in that sample. And so someone might ask, well, why not just use that all the time instead of eDNA, instead of qPCR? It has advantages and disadvantages. So the advantages are you have one assay for many species, and that's a big advantage because you might not think to test for all the species that are in that sample, or there might not be qPCR assays for all of the species that you want to test for. And so you can find species that you didn't think to test for. And it has disadvantages as well. The results can be difficult to interpret. Like sometimes the species match equally to two different species, and I have to try to interpret that bioinformatically. It can be more expensive. And right now, the turnaround time for metabarcoding in our lab is not good. It's probably 4 months or so. Whereas with qPCR is going to be a shorter turnaround, more like a month or so. Or we could try to rush things faster than that. But I've applied for funding to get a sequencer that I will be able to do metabarcoding in-house. And if I am successful at that funding, then the turnaround time will get much better for metabarcoding. Okay. Brad asked me to talk about prices too. So we do this in a lot of different ways. Feel free to call me up. I'm always happy to talk, and no obligation. Just talk about what the options are for eDNA for your specific to address your watershed or your problems. So that's no problem. And then we, sometimes help people write grants so they can apply for funding. We also just do fee for service. So people want this many samples, and we run the assays. And generally that costs about $100 per water sample for a single species, and then $25 for each additional species. Metabarcoding is more like $3,300 because we have to buy like a big lane of sequencing. You can't just do a little bit of next generation sequencing. You can only buy it in fairly big chunks. So it's $3,300 and then $85 a sample, but we can put up to 92 samples on that same lane. So depending on how many bottles you want to test, it might actually be kind of similar if you had 92 samples to qPCR, because this incurs an additional expense for each additional species you want to test, whereas this is going to capture all the species that our primers can capture. So that's the difference with that. We also do offer eDNA field sampling training. So that's when my lab manager will go out to the field with a group of people, up to 10 people, and train them how to properly and without contamination sample eDNA in the field. And it's about a half a day or so of training, and that's $200 a person. And UNB, I should say UNB likes to charge overhead on all of this, and for the sample running, they charge 10% overhead, but for the training, they charge 30% overhead, and we have to pay for our travel with that. So that's that. And then we also offer eDNA field sampling certification, and that is simply when the bottles that the students use for the eDNA training in the class are then run and assayed in our lab to confirm that there's no contamination, and the species that we know are in that body of water are present there. And then we give you a certification form that certifies you as an eDNA field sampler. And we also make sampling kits for people. And that just, I can't really put a price for that because it's just too variable depending on what exactly you want to do and what and how much you want. So we'd have to talk further about that. So those are the eDNA services offered at the lab. So we have these species that are assays that are up and running. We have the reagents. They're good to go. So they're just in alphabetical order here. Most of these are fish, but there is the brook floater there that we have an assay running for. So that's one list, and then it continues on to the next slide. So I didn't, yeah. So this just continues the list. And again, most of them are fish. We do have an assay for wood turtle as well. So that is the assays that we have up and running in the lab. But if your species of interest is not on this list, I'm more than happy to look in the literature and see if there's something available in the literature that we'd be able to get up and running pretty quick. If there's not, then we'd have to discuss assay development, which is more involved and more expensive. And now I'm going to describe a couple of the specific research projects that we've done in the lab, and both of these actually focus on using eDNA to measure Atlantic salmon abundance, which is not something that is, so presence absence is fairly well established with eDNA. Measuring abundance of individuals is not. So this is us. This is my lab and my students that are pushing the field of just seeing how far we can take eDNA. And so this specific project was a collaboration with University of Maine, as well as two factions of the DFO. And yeah, our main question is, could eDNA be used to measure abundance and distribution of salmon? A lot of that's what I've already said right there. But needless to say, it would be very useful if we could reliably measure abundance of Atlantic salmon with eDNA, because there's a lot of effort put all over to count the salmon. And so if eDNA could do it easier, then setting up traps, setting up weirs, electrofishing, all of that is extremely labor intensive. So there's great promise if we could supplement that or even substitute some of that with eDNA. So the questions we addressed: how many fish can be detected with eDNA, or how few fish? How far downstream is the signal detectable? How is collecting midstream different than collecting on the banks? And do greater quantities of fish produce a stronger signal? What environmental factors affect eDNA, and what is the ideal sampling scheme for Atlantic salmon? So this is just a schematic of what you would expect in a river. So this is a river, and the flow is going this way with the arrow. You've got a fish right here, and that fish is going to have an eDNA plume that comes off of it. And so right next to the fish, the plume is going to be very narrow. So if the fish is in the middle of the river, there won't be any on the banks, but then it's going to start to get mixed in with fluid dynamics and the way the water in rivers moves. And at some point, it's going to be much more homogeneous in the river. And so the question, we really wanted to know how this actually looks and how long the signal persists in nature. So we set up cages with salmon in rivers that don't have any natural salmon. And so we did a field experiment. And so we had various numbers of individuals in the salmon. We either had 3 for low density, 10 for medium density, 30 for high density. And then we did a very systematic sampling downstream, one upstream of the river, and then very systematically sampled downstream, both at both of the banks as well as in the middle of the river. And we took environmental factors as we sampled, which included temperature, water discharge, pH, turbidity, salinity, and chlorophyll. And this is some of the results right here. So there were 3 different rivers in southwestern New Brunswick, and 2 different times of the year. And so you can see this figure. The cage was right here in all of these figures. This is the sampling month. This is how many fish were in the cage, 3, 10, or 30. And there's 3 different rivers, and the main these arrows tell you the velocity of the river at that time. And this panel at the top is detection rate. So this is, we ran 4 replicates, I believe, in the study for each. We ran 4 qPCR replicates in the lab for each sample. And then I'm sorry, there were 3. So this tells you if 0, 1, 2, or all 3 of those 3 produced a signal. And then this panel down here is when we estimated the eDNA concentration based on the results. So a couple observations. First of all, even having 3 individuals, so here and here, we certainly detected an eDNA signal downstream. It's pretty light in this one right here, but in this one, just 3 individuals produced quite a signal that traveled quite far. So this is a logarithmic scale. So this is 1,000 meters downstream of the cage right here. And so that's the first observation. The second observation is the signal is much patchier along the banks compared to sampling in the middle of the river. So I guess those are the main take-home messages of this slide, is that it's a persistent signal. And you look at some of these that have 30 fish, I mean, 1,500 meters downstream, there's still a booming signal for Atlantic salmon. So it was quite enlightening. And here's another graph. And so this is just somewhat another way of showing the same data that was on the previous slide. So this is the distance downstream in detection rate and eDNA concentration. And so this shows you how the signal decays as you go downstream. So in different velocities. So low velocity river, the signal was higher and then decayed pretty slow. And a high velocity river, you know, you're going to have more of a dilution effect there. And these are all sampling in the river, and these are sampling at the bank. So if you look at these, right after the cages, there wasn't much signal at all right at the bank in a lot of cases. But then as you go further downstream, you see an increase in the signal and then it decays. So that's the main thing that I wanted to point out with this slide. But this was pretty revolutionary work. People haven't, we were the first ones to do it, put salmon in cages and monitor the eDNA signal. And it was great to work with DFO on this, particularly Anaïs Lacourcière and Nelly Gagne, who were leaders of the project. So and this was published in Frontiers in Ecology and Evolution. It's open source and it's freely available to anyone that wants to read it. Published in April 2021. All right, so now I'm going to shift to another of our research project, and this is so Anaïs Lacourcière is also involved with this one, as well as Dr. Kurt Samways, who's a assistant professor and Parks Canada research chair in our department. And then Melissa Morrison is the master's student that has since graduated and moved on to other things. But she's the one that actually did the work. And so this was in Fundy National Park and the Upper Salmon River. A lot of you know where that is, but here's a map just to show you, and right there in that square. So the Upper Salmon River is, geez, I think about, I want to say 15 kilometers of fish passage, and then there's some waterfalls, so the salmon can't get above there. But this, you know, this is part of the inner Bay of Fundy Atlantic salmon, which are highly endangered, are having very radical intervention policies and strategies to get their numbers up. So there's a lot of work going into increasing the amount of salmon in the inner Bay of Fundy. So yeah, this had various parts to it. I'm going to focus exclusively on the smolt in the interest of time. So smolt are in the river in the spring, and then they all leave. They go out to the ocean. And so that creates a fairly nice natural experiment for an eDNA study, because there's a smolt wheel at the mouth of the river that estimates the population as they leave. It has a catch efficiency. And so we can, at the end of the season, we can estimate how many smolts were there. And there's the smolt wheel right there. I think there's a little video for it, maybe, maybe not. Okay, it's not working, but that's okay. It's that's the smolt wheel, and percentage of them get trapped in that as they're leaving the river, And then so we can extrapolate based on that trap efficiency to estimate the entire, all of the individuals of smolt that were in the river at the beginning. So we sampled near the smolt wheel 2 times per week for 6 weeks. And the idea is we captured, we started before the outmigration started, and we kept on sampling until they were all gone. And so because we know the efficiency of the smolt wheel, we estimated that before they left, there were 4,911 smolt that were in the river. And we also took lots of variables because we're still at a space in eDNA where we don't know what is going to affect the eDNA signal. Because we want to measure fish abundance, we hope that fish abundance will affect the eDNA signal because that's what we want. But then there's also all of these other variables that could muddy the water and get in the way of interpreting just the raw eDNA signal and correlating it with fish abundance. For example, temperature has a big potential to influence the eDNA. First of all, higher temperatures are going to cause more rapid eDNA degradation because higher temperatures in general cause DNA to degrade faster. However, higher temperatures also increase the metabolism of fish. And so fish with increased metabolism might produce more eDNA as well. So it's not at all clear how temperature is going to affect the eDNA signal. It's not clear yet in the literature. So we took all of these variables, including chlorophyll, mean temperature squared. I won't get into why in this talk. The removed smolts and the total smolt abundance. So we knew the total smolt abundance every time we sampled from eDNA because at the end of the year, we knew how many total smolts were there. And we also knew how many were leaving. We could figure that out at the end of the sampling season and then extrapolate back to each of the sampling time periods. And so this is the so this is the data. This is actually the same slide here, just with highlighted, but temperature had a big effect. So the higher the temperature of the water, the more eDNA was there. Fortunately, total smolt abundance also had a big effect, and that's like our variable of interest. So we were very happy to see that that variable had an influence on the concentration of eDNA, because as we refine this method and test it in more places, the idea is that as long as you take the temperature of the water each time you sample, we should be able to correct for that and then therefore get a more accurate estimate of the total abundance of fish with the eDNA sample. So, and yeah, it had a nonlinear relationship, but I'm not going to actually go into that level of detail for this talk. And that is the end of my talk. So these are a lot of my funders, and I'd be happy to address any questions that you have.
I have a couple of questions. The qPCR slide that you showed, that showed the graphs increasing and then slowly coming together, I think it was the fluorescence graph. What species was that showing actually? when you actually take that slide?
I actually I don't know, but they all, so yeah, I paraphrased a little bit. So this is actually showing a dilution curve. So we always have a dilution series, you know, so we take a sample of known concentration and then we dilute it 1 in 10, dilute it 1 in 10, dilute it 1 in 10, dilute it 1 in 10. And so this is showing the various concentrations, because wherever each of these lines meets this line right here, you can then extrapolate back fairly accurately to what the original concentration was of it. This is an extremely sensitive assay. This is the exact same equipment that is used for COVID testing.
Okay.
Identical. It is identical. Okay. So we have the exact same machines. The same machines that are used to detect eDNA are used for COVID testing. And it is so sensitive, it can detect single molecules of DNA.
Wow.
At the beginning of concentration. And that's exactly what you'd want for COVID testing too, because that's also at very low, can be at very low concentrations. And so we're actually really glad that we bought these machines before the pandemic, because they weren't available for quite a while. So yeah, that's what this is showing. This is actually showing a dilution curve. I'm not even sure what species it is.
Okay. The assay that was used would tell you what species, right? Sorry to interrupt you.
I can't tell you what species, but they all, when you're looking at this, they all look the same. Yeah. So you wouldn't be able to look at this and say it's one species or another. Okay. The way you know it's one species or another is you know what species specific ingredients you put into the chemical reaction. So.
All right. I have a question from Jeff Baston. How has improvement in computing power enabled this level of detailed DNA assessment and assay? So, computing power question.
So that's a very interesting question. Overall, eDNA analysis is not that computationally demanding.
Okay.
However, one thing, well, the internet and online resources has been critical to the development of eDNA, because all the species in the world that people have sequenced are in GenBank and other various online repositories, and that is essential to eDNA studies, because you need to know what the sequences are for every single species that you want to test. And generally, this has already been done by somebody else. And so that's how we actually design primers often for a new species, is that we get we find sequences for the barcoding region for the species we're interested in and closely related species, and that's how we design a species-specific assay. So,
okay, so it's the sharing of worldwide, really, global repositories of scientific data related to.
That is correct.
DNA analysis of the species we're interested in.
Right. So the actual analyses can mostly be done on a laptop or a desktop computer. And that's unlike other projects in my lab. I have a huge server that has 2 terabytes of RAM, but that's used for genomic studies when we're, doing an assay scanning entire genomes of populations, not for eDNA. So.
Okay, another question from Ron Arsenault. In the slide with the vertical graphs in the 3 rivers where salmon were placed in cages and eDNA was done downstream of the cages, there were squares colored red upstream of the cages, at least as I understand the graph. What was the reason for the color upstream?
Yeah, we're not exactly sure about that. There could have been some swirling around, or there could be some salmon in those rivers that that we don't know about. So we're not really sure about that. It didn't happen very much, but it sure did. That was a pretty astute observation.
Yeah.
I'd actually have to go. We did address that in the paper. It's just been a while since I've read this paper. I'm going to have to go back and look at that.
Yeah, they don't all show it, but there's certain ones that
Yeah,
do.
there are. Yeah, that's an astute observation.
Okay, thank you, Ron. There's the answer is we don't know, but you can read the paper and find out. Well, I guess that's a fair statement. Yeah. How would you collect? This is from Dinah Stalker. How would you collect samples in a lake? Would you do it from the shore, from a boat, or?
Right.
What would be the challenge there? Or
so yeah, so lakes, everything's challenging, but lakes are a very different beast from rivers because you don't have this linear flow. You know, whenever you detect something in a river, you can fairly safely say that, okay, we detected a signal and those fish must be here or above. And so you can't say that with lakes. And lakes, you have to think about the time of the year, if the lake is stratified or not. If your species of interest are below the thermocline, then you have to sample below the thermocline. And we do have a sampling device to do that, that can get a sample at depth. And sampling from shore can be done. Sampling from boats can be done, although again, you wouldn't want a boat that you do other fish work with, or a fishing boat, because the boat itself can emanate an eDNA signal that would be not what you're trying to detect. And so, yeah, I would say that there's still a lot of active research on how to sample lakes. My advice would be to take a lot of samples, like 20 or so. With Grand Lake, that would be, you know, a whole different ballpark because it's huge. But more of a normalish sized lake, you know, 15 to 20 samples around the lake is going to get you a decent picture. If you do it in the spring, there won't be a thermocline, and so everything should be mixed. So that would be a good, just shooting off the hip kind of advice there.
Okay.
So
the cost for that would, if you took 20 samples, let's say, would be 20 times, I forget the cost, 100 plus 5 times 19 or something.
Yep.
It's going to be $500 or $600 for those 20 samples, or more maybe.
Yep.
Yeah. Okay.
Yep. And so again, that would be, you know, it's hard to make advice on just a hypothetical situation like that, but that would be, we do lots of consulting, and I'm always happy to have a conversation with people about their specific situation and advise a sampling strategy.
Okay, that's great. Thank you for offering that. I know a lot of people around the environmental communities will be glad to hear that. So I don't see any other questions from participants, but I have a couple more if you're okay with me asking in a few minutes.
Yep.
The inner Bay of Fundy salmon species, I guess, is a different species from the non-inner Bay of Fundy species. Is that correct?
They're all Atlantic salmon, so they're the same species, but they are a different designated unit, so a different population, and they have a different species at risk designation. So the outer Bay of Fundy Atlantic salmon are not considered to be endangered, I believe.
Right.
Designation. Yep.
Right, but so I guess there would be no appearance of outer Bay of Fundy salmon in the inner bay when you're doing your research.
Well, there certainly could be, because Atlantic salmon stray, and so they, you know, I wouldn't exclude that there's not movement there.
Okay. All right. So that's interesting. I didn't know that was possible. The other question I had concerned weirs. We have quite a few weirs in the Jemseg Grand Lake watershed and the Canaan, Washtenamoick watershed, other watersheds in the St. John River. So would you be able to take samples from inside of a weir and determine what species were there? Like, were there Atlantic salmon? Were there striped bass? Just Gaspereau? Or
You mean inside the trap of
inside
the
the trap? That's correct, because they just go around in a circle. They're harvested every day or two, as I understand, so they don't die from going in circles. But I think the idea is, well, just like to know if it's possible to get a sample from the weir, inside the weir, and determine what species were there at some point in time.
Well, you could certainly sample downstream of the weir trap and assay that water. I guess I'm a little bit confused by your question, because why wouldn't you just look at the fish that are in the trap and identify them that way?
That's a good question. In the lake, in Grand Lake, there's about 9 weirs or so used every year. So the notion of downstream doesn't come into play as far as I'm aware. There probably are some currents, but usually wind.
Okay.
Yeah, but the notion of putting a camera inside the weir and looking, I guess that does make sense. You're right. Yeah, probably a lot.
Right. Yeah, that might be a better idea. Usually eDNA is used, when you don't know where the fish are.
Yeah.
And but once you have the fish, in a trap, that's, kind of done the work for you. So, yep.
Okay, got it. Very good. A couple more questions. Michael Gilstrom asks, you focused on vertebrates. What are the outlooks for using eDNA for monitoring invertebrates and plants in lakes and rivers?
People are still trying to figure it out. Even with wood turtles, we're finding they don't produce nearly as much eDNA as fish do. Mm-hmm. There was a study that I was just looking at tonight. Obviously, lots of people would like to use this to detect invasive aquatic plants like Eurasian milfoil. There was a study at Washington State that found they could detect Eurasian milfoil, but only when it was at extremely high densities. Okay. And so right now, the promise does not look good for using eDNA to detect invasive plants. As far as invertebrates, I guess I'm not super familiar with that body of literature. I would guess that they probably don't produce a lot of eDNA, but I don't actually know about invertebrates.
Okay, great. Thank you for that answer. Debbie Peck asks, why does a species have an assay that can be purchased and used for testing comparisons? Are there any agencies involved in producing a wider variety of assays?
So this Genome Canada group that I am involved with, one of the main goals of the grant is to make eDNA assays for all freshwater fish of Canada. Mm-hmm. The way qPCR works, though, each reaction is species specific, because this is literally the data that you see. Did you get a signal in the reaction or didn't you? So there's nothing that you can, every species is going to look the same. The only reason you know that species A was detected and not species B is that you did 2 different assays, one that was targeted towards species A and you got signals like this, another one that was targeted towards species B and you didn't get a signal. That's how qPCR works. So it is really a species specific approach, or as long as your assay is working. That is the goal of the assay, is to make it be species specific. And then, you know, metabarcoding is the other strategy where you can get a whole variety of species.
Okay. So I guess you can purchase these assays for the species, at least the ones that you indicated. Is that true?
Right. I mean, purchase the assays. What that means is that there's companies that just design, that produce short fragments of DNA.
Okay.
They're called oligos. So we just tell them exactly what sequences we want, and those are the assays. And so you can get those sequences from the scientific literature if somebody has already developed for that species. You could ask my lab to design new assays for species that you want to be able to detect, but it's not like it comes in a box that says Atlantic Salmon eDNA studies. It's more like we have a relationship with this company. We say, design these molecules with these sequences of base pairs, and they just do it. And they do this for all kinds of people that do research in all divergent fields of genetics, not just eDNA.
Okay, I got it. Okay, so it's a complex system already, even though it's sort of just starting, as you mentioned, especially for eDNA. Ron Arsenault asks a question: can you distinguish using DNA analysis between inner and outer Bay of Fundy salmon?
So yeah, so that wouldn't be eDNA. That would be more like taking fin clips and scanning the genomes and making inferences based on population genetics. And I would have to look at the literature. I believe they are distinct, but I would have to go over those papers to give you a good answer there.
Well, it's always good to have open questions. Yeah. Okay. I guess that's all the questions I see in the chat session. Sherry Elke says, very informative presentation. Thank you. Okay, so Ron Arsenault says, Barcode of Life project, the Barcode of Life project produces a reference sequence from vouchered specimens. See genomecanada.ca. So,
yeah, and so, yeah, I mean, yes. Barcoding in general was really started and is led by Canadian researchers. The Barcode of Life project is to, characterize the cytochrome oxidase barcode region for every species on the planet, or every animal on the planet at least. That's run out of University of Guelph, and that's a great research. That is a great resource for eDNA projects because of their work.
Yeah, that's a good example of scientific projects helping basically other scientific projects move forward based on building on the shoulders of giants.
Yep, exactly.
We used to still say, I guess. Well, I don't see any more questions. I want to thank you again, Scott, for preparing and presenting this webinar, answering questions. We look forward to many more good results from your research, and hopefully we can get to use some of your eDNA techniques to advantage in our water quality observation programs. And with that, I'll wrap it up.
Well, thank you very much. It's an honor to present to this group, and thank you everyone for spending the time to take a listen.